so we can start the lightning talks of the afternoon. We will have 6 talks of 5 minutes each, so very quick. I don't know if we have any time for questions, depending on each speaker, but we'll try to make a smooth transition. So if you're one of the speakers, I'd like for you to, before you give your talk, you come closer to the stage so you can be introduced, and we actually start your presentation as soon as possible. My name is Nick Serral. From Iowa State University. I'm one of the organizers of the meeting. I hope you guys have had a good time so far. You guys are thirsty for more discussion in the evening, at a bar perhaps. Jim said that he'll be paying for the drink, no? Am I? See, such a nice guy, right? Good. So that being said, I'd like to introduce the first speaker of this afternoon, Chris Elsick. Please welcome Chris with me.
So I'm going to try to talk like lightning. So I'm going to be talking about our data mining warehouse called Bovine Mine, and this is a resource that allows you to mine for genomic kind of data from a lot of different sources, and then also data from animal QTLDB, gene expression data, variant effects, things like that. And so, this, we don't actually have phenotype data right now, but the way that I fit into this is, so Natalia earlier mentioned how, sort of like the activities that you do from collecting phenotypes all the way up to using the phenotypes to make predictions. And then there's one part of it that was like the added value part, where you actually integrate your phenotypes with other data, including genomic data. And so this is where I feel like we fit in, and what we want to do is to develop Bovine Mine. We want to develop a NoSQL extension that would actually be able to handle large, high throughput data types such as genotypes and phenotypes. So, but for this talk, I'm really going to just tell you what Bovine is at the moment. So this is the Bovine Mine homepage, and it has it shows different tools that you can use and different ways of searching. And then here is a place that lists different kinds of template queries that look like this. So we have all kinds of predefined queries that you can enter constraints. You can either enter IDs or lists of IDs in other constraints, and then you can edit the query using the query builder, and you can or just build a query from scratch. And then when you do a search, you end up with a table output that is very customizable. So you can these icons allow you to do different filtering or removing columns. You can add more columns, and you can export your data in different formats, including tab-delimited files. And then, so we have a list tool where you can upload lists of IDs, and so these could be gene IDs, SNP IDs, SNP alias IDs, transcript IDs, all kinds of IDs, and be able to save lists and do things like set operations on your list. So this is a really good tool for meta-analysis if you want to compare your results with published results. If the people provided good supplemental data sets with their publications. There's also a region search tool where you can upload coordinates and then select different genomic features within a specified range of the coordinates. So this could be SNP coordinates, or it could be regions, QTL regions or something like that. And so that gives you, that actually allows you to create a list of whatever that you selected that you can use in further analysis. So, so now I'm actually going to, I want to start a movie, but I might not because I don't see it here. Okay. No, I don't think, I have a movie that I could show maybe some other time is there? I'm not very good at, I'm not actually good at, there should be like there's actually a thing that might show up there, but maybe not, no. Okay, never mind, I'm not gonna show you a movie. So that's that's it. Does anybody have any questions? Okay.
Questions for Chris? No? Okay.
So the next speaker is Lima from University of Maryland, and he'll be talking about analytics of large U.S. dairy cattle something that I missed. Let's see, pass the slides. Genomic phenotypic databases. So please help me welcoming Lima.
Good afternoon, everybody. Actually, with so many people, colleagues from CDCV and UHC AGIL, there are many people good to present this. So I will try my best. So I want to introduce you the largest cattle genomics and phenomic database in the US. And I think, to the best of my knowledge, this is the largest across livestock species. So this is a direct cattle database with both phenotype and genotype data, a large amount of data collected. In addition, there are the pedigree and other environmental factors collected. So actually, a good variety of phenotypic records have been collected for dairy cattle, including yield, dairy production, body conformation, longevity, fertility, calving measurements. The collection of phenotypic data could trace back to over hundreds of years ago. And here are some numbers related to phenotypic records. Some of these numbers are outdated, but it's still good enough to show you the scale of the phenotypic data collected in the database. It ranges from 600 million daily lactation daily yield records to 20 million calving information. And this slide just shows you the scale of genotypic data. And so far, till 2017 this September, there are over 2 million animal genotypes collected in the database. And this shows you the trend addition of animal genotype per year. And in the past year, now we have about 500,000 animal genotypes collected. So with this kind of data, the Council of Dairy Cattle Breeding and USDA Animal Genomics and Improvement Lab do make good progress. This has been an example of a success genomic selection and genetic selection in livestock species. So these are just some evidence showing the constant increase in cattle production and dollar value of the dairy cattle. As a collaborator, including our lab from University of Maryland, we also use this database to do all kinds of research projects. But our research focuses on dissection of the relationship between genotype and phenotype. We do two directions of analysis. GWAS to find the genes responsible for complex traits. We also do prediction of breeding value and also phenotypic value based on the genotype data. In particular, we try to incorporate the sequence information now into these two types of process, and we also want to incorporate the new traits added. It has been very many successful stories involving this database, but there are tremendous challenges, particularly in computation side. Current evaluation or calculation of breeding values takes weeks to finish one round of analysis, and that's only using 60,000 SNPs. And thinking about millions of SNP data in the sequence, so if we want to include more SNPs into this calculation, and considering the 2 million or more in the near future animals to be analyzed, this calculation is facing a large amount of computational challenge. And we are also trying to add more data into more animals and more traits to this database. So in summary, it has been a successful program in dairy cattle to do genetic selection using this big database. And there are a lot of research opportunities and also challenges associated with using this database. With that, I want to thank our collaborators and funding agencies. And any questions you may have.
So thank you, Lima. Any short question to Lima? Well, if not, let's thank Lima once again. So our next speaker is Brittany Keel. She's from the USDA. She'll be talking about incorporating deep learning into the analysis of diverse livestock data. Please help me in welcoming Brittany.
Okay, well, I should start by warning you that I gave the title of this talk before I knew it was 5 minutes, so we're by no means going to go over all of deep learning and how it can be applied, but I'm just going to give you a really quick snapshot. So first off, what I mean by deep learning is a subset of machine learning that basically focuses on learning the relationships between data inputs rather than more of a task-oriented algorithm. And I want to talk about one particular type of deep learning today, and that's a neural network. So just to get started, what is a neural network? So an artificial neural network is a machine learning technique that's inspired by how neurons in the brain function. So you can think of information coming in from multiple sources in the dendrites of a neuron, comes into the cell processes that information and sends an output signal down the synapse. So it's a similar type of thing. Artificial neural networks are composed, just like the brain, of a large number of highly interconnected neurons. This is just a snapshot of one type of neural network. So this is a feed-forward neural network. The way this works is there are 3 layers. There's an input layer where the signals come in. There's a hidden layer, which are the processing units in the neural network, and then an output layer. So between each of those layers are these edges in the network graph. So those are sort of those are the dendrites coming in. So these are weighted connections in the graph, and the weights for these connections are learned from a set of training data that the user would input into the model. And so then the idea being you train up your neural network, and then you want to use that for the prediction of something later. So you can put in a set of inputs and get an output prediction. So there are a few advantages to this type of modeling. One of them is that neural networks have really good ability to learn relationships between really complex data, so things that are highly nonlinear or imprecise data. And what I mean by imprecise, maybe missing data points or just noisy data in general. And neural networks actually require a pretty small amount of training data compared to other modeling instances. These are very easy to implement in parallel, so you can actually utilize GPU computing or multi-core processors, so you can speed things up quite a bit. And they have, the really big selling point here, I think, is the ability to achieve a high level of learning with pretty low supervision. So I was looking up just what applications of ANN are out there in livestock, and it's actually pretty limited. So there's been some prediction of yield traits in dairy cattle, a little bit of work in breeding values in dairy and Angus cattle as well. And then our group has been working on predicting feeding behavior in grow-finished pigs based on thermal conditions, and we're using an RFID technology to monitor that feeding behavior. So I've got that highlighted in red. We do have a poster right over there if you want to go check more of that out later, and Tammy Brown-Brandel will be up here pretty soon, and she's going to talk a little bit about that RFID technology. So just to kind of wrap up, just future applications and where I see these types of models being used. The first 2, I'm sort of working in that space. The last one, not so much. So the incorporation of multiple types of omics data to predict phenotype. So we generate proteomic data and genomic data, transcriptomics, metabolomics. How do we piece that all together when, those relationships, we don't actually know what to expect as far as a functional form goes, so this could be a really useful tool in that space. Also, the development of real-time behavior monitoring systems, and that's what we're working with on the swine side with that feeding behavior. And then I can also see this working well maybe in calculating breeding values, especially for multi-traits, where you've got probably very complex relationships, often not linear, and this would be a good use there as well. So with that, I'm going to go ahead and take any questions you have, and I can also talk later if that's better.
Have time for one quick question? Yes.
Hi. You mentioned that you don't need a big training data set, and also that neural nets don't need too much supervision from user. And then you have a deep learner, it will be a deep network. So our experience is the opposite, is that this is such a powerful tool that tends to overfit the data. So if you don't supervise, don't be careful, you are going to overfit and maybe copy some noise. So the two issues, you either have a bigger data set or you be very careful with overfitting. So what is your experience with this then?
Sure. So I think the training data is very important, getting the right training set, but also there are many, many types of neural networks as well. So that structure that I showed you on the slide, that was just one particular type. So there are different options for the type of problem that you're working on, and so potentially the overfitting might be something that's going on in the actual training or the structure of the network as well. So there's some considerations there that may help resolve some of that. But I do agree, they can overfit, they do have that tendency, but as long as you're doing it in the most correct way, if that makes sense. So yeah, I think it comes down to understanding the type of problem that you're working on and which model is going to work best.
All right. Thank you. Let's thank her once again.
Thanks.
Our next speaker is JP Steibel from Michigan State University. I'm assuming you're still gonna talk about pigs as well?
Yes.
Matrix, see? Incorporating of behavioral observation for the estimation of social genetic effects. Please help me welcoming JP Steibel.
Thank you. Thanks, Nick. So I think all of us are, or most of us are familiar with direct genetic effects, which is the portion of the phenotype, the phenotypic variation that is due to inherited genetic effects that are presumably in the DNA. So that's an effect of the genetics of an individual on its own, the expression of its own phenotype. I am now interested in modeling the effect that a genotype of an individual has on the expression of the phenotype of the social group mates. And for that, there's models already that have been proposed. I don't know if there is a pointer here. There you go. Thank you. And if in a very simple case, if all the individuals in your relationship matrix have phenotypes, have observed phenotypes, the incidence matrix for the direct genetic effect is just an identity matrix. And in the case of indirect genetic effects, in this case, social group effects, we need to include here a incidence matrix that will look somewhat like this. Imagine that in this case, in this cartoon example, I have two social groups. This is the animals that correspond to one social group. These are the animals of the other social group. The first thing that you notice is along the diagonal there are zeros, because we are discounting or not counting the effect of the genetic effect of that individual on its own phenotype, but on the phenotype of all the group mates. And this is the typical assumption that there is a uniform interaction matrix between all of those group mates. Of course, this is just a null model, and it's adopted by convenience more than by sticking to reality. We know that that's not the case. So to cut to the point, what we have been working on at Michigan State with my colleagues Gianni Sigford and Kathy Ernst is the estimation of the individual interactions between all animals in a pen or in a social group. For example, this is a matrix for a group of 13 individuals, a pen with 13 piglets. And what we have here is the number of seconds that each pair of piglets spend interacting aggressively with each other. And there is different types of interactions. For example, reciprocal fights, attacks, single bites, inverse pressing, parallel pressing. These are behaviors that are precursor of an aggression, head knocks, etc. And our proposal is to produce these different types of interaction matrices based on different behaviors and use these to parameterize social group effects for several traits. For that, and this is my last slide, the limitation for that is the automatic recording of all possible dyadic interactions between individuals in every possible social group that an individual was housed in. So for that, of course, we have a gold standard, which is a very rich data set that involves several dozen pens, about 1,000 pigs, mixed and remixed, and a small army of undergraduate students that have been doing the decoding supervised by a few graduate students. And so that will be our gold standard. Our next step will be to move to automatic recognition of behavior using video, and I think there's other groups interested in the same idea. The first, number one challenge is this, individually track each animal throughout their interactions in the pen. If we can get that done in a robust way, with that alone, there is a first matrix, which is a matrix of proximity, which is a proxy for the matrix of aggressive interactions, but also could be a matrix for non-aggressive interactions. So that will be kind of noisy. The next one, once the individuals are tracked, will be the inferring the interacting dyads from those trajectories, and then inferring which of those interactions were aggressive based on several different patterns of movement. We also plan to overimpose on these other layers of information. For example, we'll have thermal cameras synchronized with these video cameras, and these thermal images will capture a heat signature of animals that were just fighting. And our colleagues, our co-directors of this grant in Scotland Rural University College have a very good data set for calibrating that. And finally, we plan to use also some wearable devices. That's it.
So I think you heard Jim's cell phone, right? So because of that, please save questions to JP Stabile for later and help me thanking him for the talk. So our next speaker, Jennifer Woodward-Green. She's from the USDA Beltsville. And she'll be talking a little bit, change gears a little bit. Well, her title is Down on the Farm, Getting My Goat, and Nothing Else. Well, that's pretty different I've seen so far. And digital phenotyping, computer vision for digital phenotype measures in livestock. Please help me welcoming Jennifer.
Thank you. Yeah, again, short talk, so snapshot will be what I talk about. As the previous speaker said, I have a poster over there with more detail, and I'll be around if you have questions beyond that. But I'm going to talk about getting my goat and nothing else. I developed 2 livestock digital phenotyping methods along with a simple image collection protocol that any farmer can use. The digital phenotypes include morphology or size and weight, coat color and pattern, a FAMACHA anemia score as an indicator of health or parasite status, and animal age based on teeth. My work is part of the USAID Feed the Future Livestock Improvement Project led by Kurt Van Tassel of the Animal Genomics Improvement Lab here in Beltsville. Body weight is a critical phenotype for livestock, important in production, sales, marketing, and more, so I focus there first. The digital methods developed for size and to predict weight perform equivalent to current manual methods, but they also provide digital records in real time during the measuring process. A main objective was to make the digital phenotyping accurate and farmer-friendly. So the technology needed to be simple, cheap, and accessible. The image collection is simple, requiring just 1 rear and 2 side poses and 1 close-up each of the eye and the teeth. The toolkit shown here costs about $300, and everything is available at a local hardware store except for the calibration signs. And it's accessible. A farmer's smartphone or digital camera will work. The easiest place to take a photo of a goat is in its natural settings, whether that's inside or outside. Unfortunately, natural settings pose challenges in image analysis. So even with the AdaptMap image collection procedure, the images can vary widely in terms of their resolution, lighting, and even the posing due to the environment, the camera, or the skills of the photographer. Controlled settings or structures may be more accurate. However, multi-cameras or fixed cameras or other high-tech camera or sensor solutions may increase cost and complexity for users. Still, for digital measurements to be of any value for phenotypes, the isolation of the goat and the sign seen here must be precise. The existing grab-cut algorithm for isolating image features was designed to handle natural settings, so it's an obvious choice. So let's see how well it works for digital phenotyping compared to my method, AdaptMap. Here you see an original image. This will be processed by AdaptMap, which incorporates GrabCut, and also by GrabCut alone. To handle the variability in the natural settings images, both of these methods require user input to get the job done. Here you see the output images from AdaptMap on the right and GrabCut on the left. The two images look very good. There's nothing left but goat. Either would look great on a greeting card. So what's the difference? Finding the edge, or the contour of the goat, seen in yellow for each process, shows GrabCut on the left leaves a messy edge. This is no problem unless, of course, you need precise measurements to determine phenotype. The AdaptMap method on the right is very clean and a nearly 20% improvement over GrabCut. So what's up next for AdaptMap? Full automation and several additional phenotypes shown here are in the works. These may lead to faster, high-throughput digital phenotyping in livestock. Consistent digital phenotypes collected from various natural settings may enhance multi-level analyses and comparison of breeding programs or populations, digital data sharing, and farmer adoption of state-of-the-art technology and development of on-farm health and production management tools. And I thank you and ask if there's any questions.
We have time for one question. No questions?
All right. Then I'll sneak in. There's the measurements on the final image.
Thank you, Jennifer. So our last speaker, also Jennifer, and Jennifer Smith from the Medical College of Wisconsin. And her talk is Phenominer, a flexible system for standardizing quantitative phenotype data. So please help me welcoming Jennifer.
I'd like to thank the organizers for allowing an outlier like me to talk to you about our Phenominer tool. I do work for the rat genome database, and much of our data is rat, but we also have data for a number of other mammalian species. And the reason why that's important will be clear in a little bit. So what is Phenominer? Phenominer is a database and a query system for storing, mining, and presenting quantitative phenotype data. Data is inputted into Phenominer the data that's inputted into Phenominer is standardized using a suite of controlled vocabularies, including the rat strain or a chinchilla source that expresses the sample. What exactly was measured is covered by the clinical measurement ontology. How it was measured is covered by the measurement method ontology. Under what conditions it was measured is covered by the experimental condition ontology. And then the trait that you're assessing by taking these measurements is covered by the vertebrate trait ontology. And the vertebrate trait ontology is not searchable yet in the user interface that you see, but that ability to search for a trait is coming. So we are big on standardization, and if any of you, well, those of you who were in the group that I was in, you know that I'm big on standardization. Standardization allows comparison across strains, across studies, across conditions, and ultimately across species, even when the source data has not been standardized. So I am not going to tell you how to do a query in PhenoMiner. If you're interested, I also have a poster over there. But this is the output of a query in PhenoMiner for the mean arterial blood pressure. And you can see up at the top that the conditions are different amongst different samples. In this case, all of the methods are the same, but if they were different, you would be able to see that in that list. The circled, I have a, yes. Here you can see that I have 2 different strains that I've selected, and if you mouse over one of the bars, it gives you information about that particular measurement, including what the strain was, what the actual value is, and so on and so forth. And then if you have a bar that has a plus on it, like these, you can click on that bar and it will give you the results for specific individual measurements. So in this case, you've got the number of animals. It gives you the distribution of the number of animals that have blood pressure measurements in particular ranges. And so you can see whether you have almost all of your measurements are all in one place and there's one that's an outlier, or if they're fairly evenly distributed. So again, the advantages of standardization: it allows users to compare measurements across studies, strains, sexes, ages, methods, and conditions. And this is the output in a tabular format, where you can see that you have 2 different studies, you have several different samples, you have, it's all the same measurement, and in this case it's all the same the same method, but you have different conditions. Some of them only have 1 condition, some of them have 3 conditions, and each one of the conditions is given an ordinality, so you can tell whether they're applied successively or they're applied all at the same time. So if, for instance, the controlled sodium content diet and the drug were applied at the same time, they would both have an ordinality of 1, and then that 3rd condition perhaps would have an ordinality of 2. So you can tell how your conditions were applied. So you're wondering why you would care about how we store rat data, and I want to tell you that this is actually fairly agnostic in terms of species. RGD is expanding to include data for additional mammalian species in our database, including the addition of chinchilla data to PhenoMiner. So the chinchilla data is stored using the same structure and the same ontologies as the rat data. This permits the same flexibility and ability to compare across studies, methods, and conditions. And the ultimate goal is to allow users to search across species and find a precision model for their disease or phenotype of interest. And with that, I say thank you.
So if you have any questions to Jennifer Smith, please ask her after we're done with the day. In order to keep everything on track, we're going to continue with our program, but let's thank again all the speakers of the Lightning Talks.